مساء الخير. اهلا بيكم في الورك شوب النهارده. انا لبنى سعيد، اسوشيت بروفيسور في جامعه النيل الاهليه وكو تشير اوف اي اس. هكون ان شاء الله الهوست النهارده. الحقيقه دي الورك شوب الرابعه من سلسله Towards an Interdisciplinary Science. الورك شوب بعنوان Toward Full Smart Implementable and Healthcare Devices. الحقيقه انا سعيده النهارده عشان الورك شوب هيقدمها my long time friend and colleague دكتور احمد سلطان. اهلا يا دكتور احمد.
اهلا وسهلا دكتوره، شكرا.
دكتور احمد سلطان currently an assistant professor at Nile University Egypt. He was a research associate and CAD specialist with the School of Engineering at Newcastle University of UK. He was also an R&D manager for a LED company at Qatar for 1.5 years. His current research interests include smart energy harvesting system and power management for biomedical implantable devices and lab-on-a-chip systems. He's also interested in the thermal impact of the implementable devices on human tissue embedded system design for lab-on-a-chip. Dr. Sultan research aimed to establish a new healthcare monitoring system and diagnosis on the fly by development of autonomous devices. Dr. Sultan could publish more than 60 papers in prestigious journals and impacted journals. He got the award for the best thesis from Cairo University for 2014. He got the State Encouragement Award in Egypt at 2019 for his contribution in the field of biomedical research in Egypt and worldwide. He won a fellowship from the Royal Academy of Engineering and Leaders in Innovation, and he's a fellow from the Higher Academy of Education in UK. Fadal Dr. Ahmed, we are really happy to have you today for this session.
Okay. الأول أنا سعيد إن أنا بعمل برزنتيشن النهاردة في الإياس مع دكتورة لبنى. ده شرف ليا كبير جدا إن أنا أشتغل معاها في البرزنتيشن بتاعت النهاردة. في البرزنتيشن بتاعت النهارده احنا هندي بريفلي كده البايوميديكال فين، يعني شغل البايو الكترونكس بيزيكلي موجوده فين والادفانسز والناس بتفكر ازاي في الاتجاه ده. هنبدا الكلام بتاعنا. الاول انواع الاستيميوليشن خلاص، والاوبتوجينetics اللي هو الفيوتشر تكنولوجي بتاع البايوميديكال ابلكيشنز او one of the most promising future technologies for bioelectronics. لكن ندخل على different applications or different devices such as brain machine interfaces, and one of the projects that I've worked in is CANDO project and Optoneuro project for brain interface. Also, I have another project for SelfCare projects, which show a wearable device for healthcare monitoring. And finally, I'm going to talk about how to take these devices from just normal healthcare devices to smart healthcare devices, not only for monitoring health condition, but also for monitoring themselves. هنستارت with stimulation and optogenetics first of all. يعني هنعرف يعني ايه stimulation. stimulation احنا عارفين معظم الاكتيفيتيز جوه المخ او النيورونز جوه البرين بتكومينيكيت مع بعضها through electrical signals. بالتالي لو انا عايز ادخل في الكوميونيكيشن سايكل دي، فانا محتاج ان انا اتكلم معاها برضو via electrical signals. ده الديفولت way of thinking basically. لو حصل abnormal behavior او abnormal electrical activities، ده بيأثر بشكل كبير على الhuman activity as well. بيدينا some disabilities such as epilepsy, Parkinson's disease, dystonia which is الصرع، الباركنسونز اللي هو الشلل الرعاش. كل دي امراض بتنتج عن الابنورمال behavior of البرين activity او abnormal behavior of البرين cells. الهدف من البرين امبلانتبل ديفايسز او البرين ماشين انترفيس ان انا اقدر اديتكت الابنورمال ريسبونسز دي او الابنورمال بيهيفيرز دي، وابدا اعمل لها either just only detection او ان انا كمان بقدر اعمل لها سبريشنز. وده الفيوتشر بتاع التكنولوجي. في طرق كتيره جدا للاستيميوليشن. اول زي ما اتفقنا من شويه ان انا هي الكتريكال ريسبونسز، فانا عشان اقدر ادخل في السايكل بتكلم مع الكتريكال ستيميوليشن او هي بتتكلم مع بعض ثرو الكتريكال سيجنالز. فالايزي واي اوف ثينكينج ان انا هتكلم معاها برضو الكتريكالي، هقدر انجكت electrical signals in a way to deliver some messages for the neuron cells such as okay, you can stay calm now, you can start to communicate to each other at the moment. So it depends on the phenomena that you are wanted to generate inside the brain. it governs the electrical patterns that you're gonna inject on the brain. It's very easy. You have some electrodes, and these electrodes are you can find off-the-shelf electrodes everywhere at the moment. You can send electrical pulses, high current pulses to the neuron cells. These pulses turn out to be some electrons. These electrons moves towards the stimulates the cells and generates the calcium protein. This calcium protein inject go inside the membrane of the cells, and by the time it generates another activity, and this activity for the electrical stimulation is just full stimulation, which means you activate the cell is very quiet, very calm, and what you can say only you can fire now, you can work now. This is what only we can what we can achieve with the electrical stimulation. And I'm highlighting with electrical stimulation, you can only do stimulation. This means we can do inhibition, which means the cells are actually active. And I want to say to the cell, just stay quiet, don't talk, don't say anything. With electrical stimulation, we cannot do that basically. For electrical stimulation, we can model the cells by kind of a capacitor bank or kind of such an R and C. R and C tank, RC tank. This RC tank is kind of you send your current through the electrodes, and this current basically charges this RC tank. And by the time this RC tank oscillates and generate an activity. So we model basically the membrane or the cells as an RC tank, and based on the resonance of this RC tank, we can inject the current on whatever frequency we are that can generate this resonance. Again, this is electrical stimulation, and this is the traditional way of thinking, basically. And there are a lot of groups around the world that are working on this technique, and we're still open for this kind of research. But on the other hand, we are also working on another kind of stimulation, which is based on optical stimulation. And this is a very promising technology for stimulation, for stimulating the cells, which is based on stimulating the cells not via electrical stimulation, but via light signals. And these light signals are basically depend on the wavelengths of the light. You can control the activity level, or you can control the stimulation level. And you need something like transducer to convert this light signal into electrical or into an electrical activity, which is called a channel rhodopsin, one of the most common enzymes or one of the most common proteins that are used at the moment as a transducer to convert light to electrical signal. So channel rhodopsin basically receives a light signal from the light source, convert it to charges, and they inject these charges by the nature into the membrane or into the cells, and in order to start the activity again. In this case, we are using a light signal. But the question here, we mentioned on the electrical stimulation that we can only do stimulation. On the other hand, do we have the same constraint here? Can we only do a stimulation, or we can do inhibition as well? So the question here also, we can do stimulation and inhibition. I will explain now how. So for the same, for the modeling case, we also model the membrane or the cell as an tank, RC tank, and we inject current, but this current here is a conversion of the light source. So we convert the current into light through the channel rhodopsin. So we have here a third-party element, which is the channel rhodopsin and the transducer. So it depends on the efficiency of the transducer, how much current arrives to the membrane, as well depend on the efficiency of the light source, how much light generated will arrive to the channel rhodopsin, and how much of this current converted via channel rhodopsin into light. Okay, so this is the basic definition behind optical stimulation. So optogenetics, for optogenetics, we all knew that brain is divided into sections, into some parts. Sometimes we need to stimulate specific part, and we don't need to stimulate the other parts. With electrical stimulation, again, this is really difficult to do it, to stimulate a specific section or a specific culture of the cells, because you inject electrical pattern and it just propagate via the pattern, via the brain. So you cannot control the electrical stimulation. On the other hand, with light stimulation, because we have this channel rhodopsin protein, we can inject the channel rhodopsin in a specific area that we need it, that we want to be affected by the light stimulation. And in this case, this is the only area that affected by the light stimulation. So here we can have the advantage of a specificity. I can specify a specific culture or specific cells for stimulation. And this is really important, and this is really unique for optogenetics. So first advantage that we can get it from using optogenetics is to specify specific cells. Second advantage here is we can not only stimulation, but also we can do inhibition. Like we can say, as we can say for the cell, you can be activated. We can say to the active cell, you can be shut now. We can shut the cells. And this is another unique advantage for the stimulation using optogenetics. So I've mentioned two main advantages. The first one is stimulation via light. You can do stimulation and inhibition. Stimulation via light gives you the advantage to specify specific cell for stimulation. And thirdly, and the last one here at the moment, is you can do stimulation and recording in the same time. For electrical stimulation, we cannot actually do both in the same time. We have a blind spot, so we inject the electrical current and we have to wait for some time due to the behavior of the electronics. It's saturated and we need to give it some time to relax, and then we start to do recording. Because we are sending and receiving using the same language, let's say, which is based on the electrical stimulation. On the other hand, if we are using light, so we are using two different languages. So we can send the command in light and we can record in the electrical stimulation, and gives you, in this case, the advantage of measuring, recording, and stimulation in the same time. And this is a great advantage, again, for the optical stimulation or for optogenetics. And that give us a very great advantage of that we can simultaneously monitor the impact of the stimulation on the neuron cells, and which is we couldn't do it actually using the electrical stimulation. Okay, first project that I was part on it was the CANDO project, and this was in collaboration with Newcastle University, and it was a kind of Newcastle University, Imperial College, UCL, and Neuroscience Institute at Newcastle. It was a great project with 10 million budget, and the whole idea of the project is to build a brain-machine interface system. And the whole idea of the project is to fabricate some electrodes, or fabricate these electrodes, or design and implement these electrodes injected inside the human brain. And the function of these electrodes is to do stimulation, to do recording, monitoring for the brain behavior. And when it detects some abnormal responses, it starts to stimulate in order to either to stimulate or inhibit these brain cells in order to stop the brain activity. So this is the first thing that we have here, is to, in this case, we need to have the main component of the system is an Optrode, which consists of the stimulation and recording parts, and control unit, which consists of the algorithm and the driving circuits for the stimulation and recording, and finally a lead, which is the cable that connects between the control unit and the brain implanted device. And these are the main component of the system, which are, again, Optrodes injected inside the brain, and control unit, which controls the response and the behavior of the system via the algorithms. And finally, the lead, or the cable, which is one, basically, it's really one of the main challenges for the whole system. One of the main challenges of the whole system, as we cannot send DC signals. I can tell this part of the project was the most difficult part of the project, is to find this lead and to design and fabricate this lead. Okay, so this is the first thing, which is the system component. By the time we know the system component, we need to know the goal. What's our purpose? What's our goal from this project? Is to do stimulation, and we are here based on light stimulation. Do recording, and here so monitoring the brain activity, and in this case we do something called LFP recording, local field potential recording. And finally, and it's one of the most common ways for detecting the abnormal behavior for implantable devices, and again interface between the control unit and the brain implanted device, and finally self-diagnostics, which is based on LED degradation and breakage of the electrodes. So this is like a 3D model of the proposed Optrode, let's say, and the Optrode was consisted of as you can see here, some electrodes for recording LFP signal, LEDs, micro LEDs for light stimulation, and every LED has its own and it's separate independent control circuit from each other, from all the LEDs. And then we have the head of the Optrode here, which consists of the control circuits. Just control circuits, not the control unit. They just control circuit to interface with the electrodes and the LEDs. Okay, and again, a block diagram of the Optrode architectures, which shows the control center control unit. And it was like a finite state machine unit to control when we can do recording, when we can do stimulation, whenever I'm doing recording, if I can do stimulation, how I can manage between both recording and simulation. It's kind of internal control system and a very simple finite state machine to control the response of the system. And this is the first part of the system and the control unit. And this control unit basically communicate with the control system, which is responsible for the algorithm, and send the commands to the control unit in order to either to do stimulation or recording or pause at the same time. So basically, control unit is the external interface to the Optrode to the control system. Second part, which does responsible for the recording, to do recording of local field potential LFP signals. And when I do recording, I get the reading and send it to the control unit. The control unit, by its rule, send it to the control unit in order to apply the algorithm. And then the stimulation parts, which are the LED, micro LED with light control. Again, we can control the light intensity in order to control the level of activity. We can control the light intensity to control either we do inhabitation or stimulation. Finally, diagnostics, which does different kind of diagnostics built-in self-LED. We can see at the end of the presentation how many diagnostic features that we actually implemented, and we have patented most of these diagnostic circuits. The second section of the brain-machine interface, which represents a real challenge because it controls the whole system response, and it has to be approved by the legacy, or has to be approved by the authority to apply it for human. It's based on the software and firmware, and it's really important here to highlight it has to follow some standards for biomedical circuits and the biomedical electronics in order to make sure that these electronics will not do undeterministic responses based on any condition. Okay, so as we know, software is always software, and it always, in most of 90%, it has some bugs. It can hang actually for the whole system. In order to guarantee not to have such situation, but again to respond safely against such kind of scenarios, we have to implement your firmware and software according to specific standard. In order to follow the standard, you have to be building a layered approach, which means you have to build your system or your firmware as in a layered system. Each layer is very independent on the other layers, and they interface to each other through APIs. So we have different layers. First of all, the 4-wire communication layer to communicate between the control unit and the Optrode. Second layer is the command, if you want to send the stimulate command or record the command. And this is a kind of intermediate layer between the electrodes or between the user and the Optrodes. And finally, the closed loop program, which does the stimulation and then does the recording. After that, does the stimulation based on the recorded signal, which is closed loop program. And this is actually the ultimate goal, is to do very independent system, do recording, and finally do the stimulation based on the recorded signal, and which is the final design purpose. And the most challenging section for most of the disease or most of the cases, because the brain activities, the main challenge for the brain activities, they are not regular. And there is no pattern for each disease. And for the same person, no specific patterns that we can follow. Not only that, for the same person, each seizure or each case has its own different pattern. So this is a challenge. You need to apply some artificial intelligence algorithms. You need to do some prediction. You need to do some detection from the recorded signal. So basically, we need to do continuous recording for our signals. Likely, we arrived to the state on this project that we already did a prototype for the device. As you can see from these photos, we have here the electrodes. So it's an Optrode, and this is a head stage, which has the finite state machines, the control circuits. And this was like a prototype bonded on a PCB, printed circuit board. You have here the LEDs, and you have in between the LEDs, you have the recording sites. And via this headset, you can control the performance of both the LEDs and the electrodes. And this is like, again, a 3D model which shows a different component of the system. And there's a kind of experimental results. So you can see here via microcontroller or very simple interface system. Via very simple interface system, you can toggle the LEDs on and off, not only on the air, because considering these LEDs will be implanted inside the brain. So it will be, we tested the LEDs inside saline, which is very equivalent on the impact on the LEDs as the brain impact or brain tissues on the LEDs. And this shows the robustness of the LEDs inside the saline and the robustness of our passivation. And this show an actual recording from the system. So we did run the electrodes, the recording process inside the saline, and we did stimulation. And you can show here kind of different activities and different recording based on the concentration. And this is a kind of initial testing for the system, which shows the initial success, basically. And we indeed went for monkey experiment or a rabbit experiment for this kind of for this project. So one of the case studies that we have the whole system here. So we have the control unit, which should be called the chest unit, which consists of a microcontroller for applying the detection suppression algorithm, the seizure suppression algorithm. And this head stage communicate via a serial port or 4-wire communication to the head stage that we have seen here on this head stage. And the head stage by turns communicate to the electrode, to the Optrode, in order to either do the recording or turn on and toggle or turn off and on the LEDs based on the required pattern. So this is kind of a prototype again, which is designed and implemented for a mice experiment. And we already did the experiment, and we have some rabbits with epileptic model, and we did some of these experiments, and you can see here we have the seizure. When we start to turn on the LED and apply the algorithm, we were successfully able to suppress the seizures. We were successfully able to suppress the seizures based on the applied algorithm. And likely all these work are published at the moment, and some of the work also are patent. So it's open for everyone to can read it. So this is one of the case study. Another case study was based on the monkey experiment, but unfortunately we still are not allowed to put it here in the presentation. The second project that we are, I was part on it was called Optoneuro, and this was with the European Union. And there's the whole idea of this project, again, is based on the stimulation using Optogenetics. But here too was based on a wearable device. So we were less strict in this project, basically, as we were only relying on a wearable device for stimulating, ultimate goal again is to retrieve vision for the people which are suffering from a specific disease, which is especially retina pigmentosa. Retina pigmentosa, it's kind of a lack of vision in the eye for the elderly people due to the aging and different kind of second effects of aging. So in this case, the optical nerve is still functional, but the rod and cones are not really functional. So the whole idea here is to inject, actually, again, a channel rhodopsin to act as a light sensor instead of the rods and cones, and send the pattern, send our pattern to the eye via the kind of specific and custom designed micro LED matrix that we have did. And this pattern will be able to stimulate or send the electrical signal to the optical nerve via the channel rhodopsin cells, not by the rod and cones. So basically, this is the whole idea of the project. So the project was consisted of image acquisition system. So the whole idea is we need to acquire the scene that we need to send it to the eye, which is basically whatever the person should see. So you acquire the scene, you apply some visual processing, such as simplification, cartoonization on the image in order to simplify the pattern. And then you send this pattern, do some kind of PWM, a pulse width encoding or pulse encoding. And after by the end of the day, you send these patterns to the high radiance micro LED display, and this matrix sends the pattern to the retina and to the retina, which is injected by channel rhodopsin. And in this case, the retina start to again sense the pattern and send the image to the optical nerve, and optical nerve, by the way, in turn send it to the brain, and the human again start to see the scene. Not really as if he was a very healthy, as if he has a healthy eye, but he will be able to figure out his way. So we cannot claim that he will say again like 100% or even 50%, but we will able to claim that he will be able to be again independent and figure out the scene, figure out his way. If he wants to figure out the main object in his way, so he will be to some extent independent person again. So the whole idea of this project was based on this high radiance display. So we need to design a high radiance micro LED display to be embedded inside this wearable glass. You can see here it's facing the eye, this blue like objects, and this micro LED display. So we were able to design this micro LED display with a 90 by 90 in a form of a matrix with columns and rows. So all what we need, if we need to turn a specific LED, we need to give the address of the row, address of the column, and immediately the LED with these addresses start to shine based on the amount of current that I'm injecting. So we have here different level of control. So we can control which LEDs that we will turn on and off, and how much light we're gonna send to the eye, or how much light we can send to the eye. And also we can send not only light pulses, we can also send a full pattern, because we have here a matrix, and this matrix was like 90 by 90 LED, and it was very challenging. So this kind of a snapshot of the control circuit, so you can control the LED amount of current as well as the row and column. So the main challenge for this is not actually the circuit design, it's very simple circuit, but the main challenge was to do kind of such high density LED, like 90 by 90, 8100 micro LED in a few millimeter by few millimeter. Okay, so that was really challenging to have such a high density LED in a very small area. So the fabrication process is the most time consuming process, and actually it took so much time until we arrived to a yield, an acceptable yield, let's say. So it based on some kind of flipped shipping. So we distribute the bonding, and we distribute the LED, and then we flip the substrate. It's kind of detailed fabrication process, but the outcome was something like that. It's like 7 millimeter by 7 millimeter, and it has like 8190 by 90 array. And we were able to address each LED of the 8000 and measure the light intensity of each LED. So this is results for this measurement. So you can see here, we were able to we succeeded to measure the light intensity for each LED by itself, okay, and also measure the efficiency of each LED by itself. And we were able to kind of find the distribution between the LED in terms of with respect to and relatively to the light intensity. In order to do that, we built such kind of setup, which was very complicated setup. So the setup basically consisted of an eye model. Okay, so we need to make sure the whole concept here is not to measure the output light from the LED matrix. We need to measure the light at the retina level. Is it enough for the stimulation process or not? Actually, it's not only enough for the stimulation, is it enough and safe? It might be enough and too much for causing damage to the cells. You know, light at some point generate heat. So you don't want to generate heat at the level of the retina cell. So you need to make sure that the light intensity at the safe level, safe level from the with respect to the retina cells. So the system consists of an eye model. Okay, this eye model consists of some lenses here. Okay, and by end of this, and all these dimensions are actually very accurate with respect to the eye, the human eye. So the retina should lies here for the human eye. So this is the actual distance for the retina, for the human cell, for the human eye. And we have here some optical system. And this optical system has the matrix, the LED matrix fixed to it. So we have the LED matrix fixed to the optical system in order to project the light pattern to the retina cell. Afterwards, we have here some microcontroller to measure and to control the patterns that we're going to display on the micro LED display. And we have a source measurement unit and to measure the amount of current against the amount of light to measure the intensity. At the back end, we have an image sensor. So this image sensor basically detects the pattern, and we send this pattern to a computer system to make sure that the pattern we are sending is exactly the same as the pattern that we are receiving. So this is kind of different patterns that we were able to generate on the matrix and actually receive it on the image sensor. This is kind of a light intensity measurement tool, integrating sphere to measure the light intensity very accurately. This is the eye model. This is a kind of the matrix, the optical lens that we are using. Okay, so this is a setup, and that was I have to say it was a complicated setup, very accurate, needed very accurate adjustment in the X and Y and Z axis in order to get such kind of image. So basically, this is the image that I was sending via the microcontroller to the LED matrix, and this is the image I was receiving on the other hand, on the last end of the LED of the eye model, and it shows that the image was very accurate as the one that we projected. Okay, this is the optical performance. We have discussed this one. And finally, so this is the second project, and we were able actually to build the whole system, implement the whole system. And we are in the phase for the human trials with some institutes in Germany, basically, and in US. And the system showed a very good successful results so far. The third project, basically, and this is my baby, let's say, I'm taking care and adopting this idea, which is SelfCare project. And this project is, I got some grants from EBSRC in UK, here in Egypt, and I'm applying for different grants again in Egypt for this project. And it kind of the future of SelfCare, of the healthcare system. It relies on basically continuous healthcare monitoring for each one, not only for the patient, or unhealthy people. It relies on continuous monitoring for the health conditions such as human body temperature, heart rate, glucose level, all these kind of vital issues, and blood pressure on the human. Continuously monitoring these kind of parameters give you a vital image of the human body. If you have this vital image of the human body, you can easily predict if there is any emergency will happen. So in that case, you will turn the healthcare system, instead of responsive system, into a predicting system, and you can even prevent the emergency before happening. So this system is based on and in order to apply actually this system, it has to be very cheap. In a country like Egypt, people cannot afford only for like 10, 20 EGB for buying such kind of SelfCare, such kind of a patch. And because unfortunately, one of the challenging at the moment is the lifetime of the patch. So the first patch that we have done, it has a lifetime of like half a day. So you can use it for only half a day and you have to remove it and use another one. We are targeting at least actually a week, but we're still working on this. So it's a whole system is based on kind of autonomous patch. In order to make it cheap, it has to be autonomous. And by autonomous here, I mean, we don't need to use a battery. So it has a kind of something called energy harvesting and the power management system in order to harvest energy, basically, in order to avoid, in order to use natural energy. So in this case, we can cut the price down. We don't need a battery, we don't need a supercapacitor, but we need to be smart enough to detect the amount of energy that we have and adapt our performance based on the amount of energy. So this is the main challenges for this device. The challenge is not the sensor, but the challenge is how to operate with no battery. So the current existing solutions is such kind of similar stuff. So one of them is painful actually. You need to use a needle to get the blood sample and then you measure the glucose. This is kind of a glucose system. Or Medtronics actually had also a kind of a wearable glucose sensor similar to this. And it costs like 50 pound, which is 1,000 EGB, 50, yes, almost like 1,000 or more EGB, which is almost expensive for the people here to afford such kind of device every, let's say, every week or every month. And this one is painful and it's annoying if you're gonna have a sample continuously every couple of hours. So we have seen a lot of people that we need to have, that they need to have kind of glucose measurement every couple of hours. And this is really annoying, really painful. So we need to have something painless, cheap, and can do this continuous monitoring based on the health conditions. And in order to do that, we need to have something called energy harvesting. Fortunately, we are using glucose to measure the glucose level, and the glucose is the source of energy in the human body. So why not using, again, glucose to be as a source of energy for the cell, for the device as well? So we're building kind of a biofuel cells in order to harvest energy for such kind of system. We need to do kind of, in order to increase the lifetime, we have to do kind of impedance monitoring in order to do how much degradation on the electrodes and then do compensation. The device, as you can see here, is based on a flexible electronics. So we have to do it in a flexible substrate. So that's another challenge here, to do it in a flexible substrate. Okay, so we have actually implemented the first trial of the device and the first prototype. It consists of a microcontroller with a Bluetooth, low energy Bluetooth communication, and as well as energy harvesting using biofuel cell. And it was able to turn on the system for a couple of minutes. And if we do continuously energy harvesting, so we'll be able to run continuously, basically. And again, we have here some electrodes, and these electrodes were actually the main limitation for the lifetime of the system. And in order to compensate for this limitation, we have used also impedance spectroscopy system in order to continuously monitor the impedance of the electrodes, compensate for the impedance change, and in this case, we can extend the lifetime of the system. So this is kind of, this is a block diagram of the SelfCare device. And this kind of also very simple, basically, power management system. So you need to turn on and turn off the peripherals or the sensors that you need based on your need, or based on the system need, or based on the different readings. And this is basically depend on some prediction algorithms that we are at the moment, and optimization algorithms that we are at the moment developing here at Nile University in the group. Okay, so we have designed again the first prototype. So this is the image of the first prototype and second prototype. So the first prototype was like a rigid PCB on a four-layer rigid PCB. And it didn't have the biofuel cell. It has just the biofuel cell interface. But the second trial, or the second version, it has everything on the PCB based on micro needles and the microfluidics to have a built-in biofuel cells and needles. So at the back end, it has only two layers. So the first layer for the electronics and the bottom layer for the microfluidics and the micro needles. And we actually succeeded to implement everything, and we were in the phase for testing it. And finally, is to do the self-diagnostic. So all what we have mentioned so far is just a healthcare devices, very normal healthcare devices. As actually, it's not really normal. It's just traditional way of thinking for healthcare devices, which means it's you implement the device, you wear the device in order to do the healthcare conditions. So all what you are thinking about how to do the healthcare conditions, but how we can guarantee that the device is still actually working. So this is the whole point here. So the whole idea here is not only to have the devices working, but also you need to make sure that the device is also working properly. So you not only need to monitor the health conditions, you need also to monitor the device condition. And this complicates the device, but increase the efficiency of the device. You don't need to use the device in a brain for 2 years, and then you open the brain, the human brain, and take the device out and throw it away. You need to implement the device on the human brain for 10 years, at least 10 to 20 years at least, and then you at this stage you can open the brain again after 20 years and remove it if you need it. So this is the whole idea. We need to do self-diagnostics for the device. So one of the in order to do self-diagnostics for these devices, you need to first list the different scenarios that can happen and different causes that can cause the damage or let's say lack of the performance of these devices. Okay, so one of the most common scenarios is the LED. For the stimulation stuff, LED is the bond pads of the LED, where the places where you can stick the LED sometimes are not very efficient. If we go back for this slide, you can find here very big variance on the LED stimulation level between different pixels. The main reason for this variance in the stimulation level is the bonding efficiency. The bonding efficiency and the bonding efficiency. So the bonding efficiency means how you fix the LED on the Optrode or how you fix the LED on the LED matrix, or how the electrodes behave basically over the time. It changes over the time. So it's very important actually to monitor. One of the very important things, the electrode, the LED bonding, if you can detect if the LED cracked or not, if there is any crack or not. Actually, this is easy. The most important thing, if the LED degrades after time, you need to detect if there is any detection, any degradation on the LED performance. So we have different things actually to detect or to measure here the anode and the cathode as the normal bonding. So there is an impedance here. We have modeled actually this is a normal impedance. It has a capacitance effect and resistance effect. And if the resistance effect increase, the efficiency of the LED decrease basically. And it causes a secondary effect, which is the heat. So we need also to monitor the heat. So one of the scenarios we need to monitor is the performance of the LED, the degradation of the LED bonding and the cracking of the LED. The second scenario is to monitor the heat, which is a secondary effect of using LEDs, or even secondary effect of using electronics. So you need to monitor the heat impact on the electronics and the heat level of the devices. Okay, I can tell some of the LEDs that I have used, it has an efficiency like 2-3% efficiency, which means more than 95-96% turn out to be a heat. Like if you're injecting like 1 watt as a power, only 2.3 milliwatt. Okay, 2.3 milliwatt of this turn out to be an actual light, and this is what you really get the benefit of. And the rest of the power turn out to be a heat, thermal heat. This is a really striking results. And of course, you don't need to burn the human eye, or you don't need to burn the human brain cells, because it's kind of, as I believe, it's kind of a very precious cells. You need to make sure that they are very safe. So you need to monitor the temperature heat generated from the LEDs or from the electronics, or from the recording electronics. We have here some actually measurements. So we have here a thermal camera to measure the light. So you can see the temperature goes up in a few seconds up to 41 degrees, just for a few seconds, or from turning it on with a full supply. Like here we have at 4 volt power supply. It turns out the temperature goes up to more than 47 degrees in 5 minutes. Okay, so this is very striking. So we need to monitor the temperature, and in this case, we have two choices. We are either to include a temperature sensor, which is actually based on a PN junction. The question here, why do I need another PN junction if I have an LED, which is a PN junction? So we have to, in this case, we can use the LED by itself as a temperature sensor by reverse biasing the LED and then measuring the current on the reverse bias LED, and this gives you indication of the temperature level, as you can see here. So this gives us indication of how much temperature of the LED by measuring the reverse current. Actually, we are talking here about the surface temperature, and this is what we actually looking for. Surface temperature is the temperature of the tissue that touching the LED, or the human skin that touching the electrodes. Okay, so we have patented this actually, and this is a kind of idea to use the LED as a temperature sensor. And this is a kind of finally a block diagram of the Optrode. Latest version of the Optrode, brain implanted Optrode, which consisted of the circuits for not only stimulating but also to use the LED as a temperature sensor, and we were successfully fabricated, we successfully fabricated this Optrode in order to do both diagnostic, self-diagnostic for the electronics, and as well as diagnostic for the human tissue, which is the main function. And this, as a consequence of this, we were able to extend the lifetime of the tissue and the lifetime of the electronics, and as well as the lifetime of the whole system. As a conclusion for the talk, medical devices have a potential impact as a future of healthcare system. Obviously, it can change the human life, can change the well-being. And so this, instead of going to the hospital every few days, you don't need to go to the hospital at all. You just go to the hospital only if there some emergency will happen. So you can predict, you can see. Okay, semi-smart medical devices which able to predict and prevent abnormal behavior of the brain activity. Fully smart medical devices, which is a new definition to not only monitoring the health condition, but also monitoring the device condition. And finally, we can achieve all of these also in terms of the power management. And thank you for all the collaborators, as well as to my university and Dr. Lubna for hosting the talk.
Thank you so much, Dr. Sultan, for this fruitful and wonderful talk. Okay, I will give 5 minutes for the audience. If they have any question, you can either post it through the chat or raise your hand and go directly to talk to Dr. Sultan. Okay, Dr. Sarah, go ahead.
At first, I would like to thank the organizers and the speaker for the very interesting talk. In fact, I'm coming from the medical background, so I understand and appreciate how valuable this idea is for the lifestyle of the patients. I do have a question, which is if those devices are to be implanted in the body, and during the discussion, there was an estimate about the lifespan of those devices. So in case of the glucose monitoring one, how long does it last in the body? And what about the fluctuation or the standards or the noise and the backgrounds? Thank you.
Okay, so first of all, for the glucose device, it's not implanted, it's a wearable device. Okay, so it's a patch, you can wear it wherever you can feel happy with it. And our goal is to last for a couple of weeks at the moment. And it's very painless. It's painless basically. So no harm at all. It's a patch. You put it in your body wherever you like, and after 2 weeks you take it out. And for the fluctuations of the glucose, we do continuous monitoring. So we build a kind of a pattern, and we use some filters to compensate for these fluctuations. So we can kind of figure out how the body behave according to the different activities. Is it clear?
Yeah, thank you for answering the question. Yes, now it's clear. Thank you.
Any other questions?
I have something that is about the future aspect. Do you think that it can be somehow achievable in the future that those devices, with the help of artificial intelligence, for example, would take the decision, like we implant along with them, or we have along with them like insulin syringe or so, and then according to the glucose level they would take an action, or it's now having the digital glucose level so that the patient itself would take an action?
Indeed, I agree with you. It can happen at some point that the device will be able to make the decision, but to be honest, I don't see this point at a near future. Like you can take, if you're talking about 5 to 10 years, yes, this will happen, but in a few years, I think this is really difficult. You know, it's really difficult to trust the electronics from the first trial. It takes time.
Yeah. In Egypt nowadays, we see ads for devices to measure the glucose level without the need for the blood injection step. They depend infrared technology.
Yes, IR. This is kind of similar to what we are using at the moment as a fast solution, but we are using also some implanted, some different technologies based on the fabrication process.
Okay. Thank you for the.
Thank you, Dr. Sultan, and thank you, Dr. Sarah, for the very nice and actually well-informative questions. Do we have any other questions from the chat? I think people are saying that the talk is valuable and it is really a nice talk, actually.
Thank you. Hopefully it was beneficial for everyone, basically.
It is because we are trying here in this years to make people interact with the concept of interdisciplinary and how this is important. And actually you are this talk is actually fit directly to the scope because you are basically electronic engineer and your playing is in our brains. So, so
your brain are safe.
Okay. So I think it is really wonderful, and I believe it will shed the light and make people trust more in engineer and the communication between engineering and the medical doctor and how we can make life better. Yeah. Okay, Iman, go ahead. You can ask your question.
Really not a question. Thank you for Dr. Sultan and for دكتورة لبنى. ونتمنى منكم مزيد من التوكس ان شاء الله. توفيق ان شاء الله. شكرا دكتورة لبنى. شكرا دكتور احمد.
Thank you.
Thank you so much, Dr. Iman. Okay. Any other questions? Okay. Thank you so much, Dr. Sultan, for this wonderful talk. And we hope that we can host you when you make level 3, inshallah, with the device and the experimental. And we would happy to try it. Thank you so much.
Thank you. Thank you.
Thank you. Finally, we hope you a very nice evening and enjoy the rest of the day. Thank you so much.
Thank you. Bye.
Bye.